Hello my dear students, welcome back. So in the last video I discussed about the cilia and flagella. Clear? Now today we are going to discuss about the centrosome and centriole. Before I start this topic centrosome and centriole, just we should have a recap of the structure of the cilia and flagella, because there is some similarity as well as some difference between the structure of the cilia, flagella and centrosome and centriole. So from the NCERT diagram of the NCERT I discussed to you. Okay, so that I discussed to you that this is the cell membrane, this is the axoneme. Axoneme has the peripheral microtubules which are in the form of doublets, 9 in number they are. Then the central microtubules, they are singlet, they are connected with each other with the help of bridge. And then the central sheath is there which will cover the central microtubules, and this this peripheral microtubules are connected to the central sheath with the help of radial spoke. Okay, with the help of radial spoke, and total 9 radial spokes are there. Okay, so all these structures we discussed. Now we can compare. So now we will see that what is the structure of centrosome and centriole. So basically centrosome is a cell organelle. This centrosome is a membraneless cell organelle which contains the centriole. Clear? So this centrosome is a cell organelle which will contain both the centrioles. Actually, 2 centrioles are there, and both the centrioles are right angle to each other. Suppose this one centriole I am discussing here. So suppose it is one centriole, then the other centriole will be perpendicular to this one. And both these centrioles, they are present inside a cell organelle, and that is called as what? These two are centriole. This is centriole. This one is also centriole. And you can see both of them are perpendicular to each other. And the space which is present around this centriole, that is called as pericentriolar material. Pericentriolar material. Pericentriolar material. Clear? So this is actually what centrosome is. Okay, so centrosome, you can see this is the cell organelle which contains both the centrioles. Here some rod-like structure I have shown here. Actually these rods are nothing but they are the fibrils. They are the fibrils which are made up of a protein that is called as tubulin protein. Okay, now we will study in detail the structure of one centriole. Okay, so either any of the centriole we can take, because structurally both of them are similar. But before I discuss the structure of centriole, some points write down. So centrosome is the cell organelle which contain both the centriole perpendicular to each other. Clear? So I told you in the surrounding of the centriole, they are covered by a amorphous substance that is called as pericentriolar material. So centrioles in a centrosome are surrounded by and amorphous are surrounded by an amorphous pericentriolar material. Clear? So as I told you, both these centrioles are at right angle, or we can say both of them are perpendicular to each other, and each centriole in a centrosome gives the appearance of an cart wheel. Okay, cart you might have seen. So it gives the appearance of a cart wheel. Clear? So that cart wheel you might have seen, a center axis is there, and then all around the diameter of that center, that means a wheel, or we can say solid wooden frame is there, that is circular wooden frame is there. So similar structure is there in the case of centriole also, that I will discuss. Okay, so both the centriole in a centrosome lie perpendicular to each other, in which each has an organization like the cart wheel. This is very important. Like the cart wheel. Clear? Now I will discuss the structure of the centriole. So if you want to understand the structure of centriole, one diagram I want to show you. So this is a diagram actually. So this is the diagram of the centriole. So you see here, now we may compare also the diagram with the cilia and flagella. Here also you can see in the peripheral sides fibrils are there, but these fibrils, they are not made up of microtubules. Rather here they are made up of tubulin protein. In the case of cilia and flagella, it was made up of microtubules, but here it is made up of tubulin proteins. This is the first difference. There we have seen that the peripheral microtubules are in the form of doublet, but here the microtubules, or we can say here the fibrils, are in the form of triplet. You can see they are in the form of, in the group of 3. So here the naming is also done, A, B and C. Okay, so this is how the fibrils, we can say, they are present, the peripheral fibrils are present in the form of triplet. So they are present in the form of triplet. And you can see the central microtubules are absent. Instead of that, here in the case of centriole, they have a proteinaceous structure that is called as hub. This structure is called as what? Hub. And this hub is made up of proteinaceous substance. Clear? Then this hub is connected to the peripheral fibrils with the help of same structure that is called as radial spoke. So how many radial spokes will be here? Here also the radial spokes will be 9 in number. You can see 1, 2, 3, 4, 5, 6, 7, 8, 9. So total 9 radial spokes are there. So if I ask you that what is the pattern of arrangement of tubulin proteins, or what is the pattern of arrangement of microtubules in the case of centriole, then that array is 9 plus 0. Very important. Here the pattern or we can say the arrangement of fibrils, or we can say proteins, are 9 plus 0. 9 are peripheral fibrils which are present in the form of triplets, and the central microtubules are absent. So we are writing 9 plus 0. Clear? So this is the difference between the structure of the cilia, flagella and the centriole. And you can see this gives the structure of a cart wheel. So this is the frame actually, and on the diameter, or we can say on the periphery, it appears like that a wooden frame is there having the same radius. Okay, So that's why this is given a special name. It is a cartwheel structure of the centriole. Clear? Now what is the function of the centriole? The centriole, I told you in the last class also, that the centriole forms the basal body of the cilia and flagella, because the cilia and flagella originates from the basal body, and that basal body is formed by the centriole. And also you should know that this centriole or centrosome is exclusively found in the animal cell. In the plant cell they are absent, and they have a very important role in the cell division. How? Actually what happens when the cell division takes place, then spindle fibers are formed, and the spindle fibers are formed from the centriole. The spindle fibers are formed from the centriole, and when the spindle fibers are formed, that in turn will form the spindle apparatus. About the spindle apparatus I will discuss in the cell cycle and cell division, but this is the function of the centriole. So it helps in the cell division by forming the spindle apparatus, and also it forms the basal body of the cilia and flagella, and it is the basal body of cilia and flagella from where the origin of the cilia and flagella take place. Clear? So now you may write down. Each centriole is made up of 9 evenly spaced peripheral fibrils of which protein? This protein, name of protein is very important. Peripheral fibrils of tubulin protein. Peripheral fibrils of tubulin protein. Then I told you this peripheral fibrils, they are in the form of triplet. Triplet means they are in a group of 3. What was the condition in the case of cilia and flagella? It was present in the form of doublet. So each peripheral fibril is present in the form of triplet. Is in the form of triplet. What is the meaning of triplet? Group of 3. So they are present in the form of triplet. So one more thing I want to tell you, that this adjacent peripheral fibril, this we can see, this adjacent peripheral fibrils, they are also connected. Like in the case of cilia and flagella, they were connected with the help of linkers. So same linkers are present here also. So here the peripheral triplets, they are connected to each other with the help of linkers. Okay, so same structure is here. Each peripheral fibril is connected to the adjacent fibril with the help of linker. So they are connected to each other with the help of linker. So whether they have the central microtubules or we can say central fibrils, they have? No. So in the center they have a proteinaceous structure that is called as hub. The central part of centriole is composed of proteinaceous rod-like structure, proteinaceous rod-like structure called, that is called as hub. So remember, this hub is present in the case of centriole only. They are absent in the case of cilia and flagella. Then I told you that the peripheral fibrils, they are connected to the hub with the help of radial spokes. The peripheral fibril are connected to hub with the help of radial spoke. So how many radial spokes will be there? 9 in number. 9 in number. Now next thing you should know that what is the function of the centriole. So I told you the centriole is very important because it will form the basal body, and the origin of cilia and flagella take place from the basal body. Means we can say it will form the basal body of the cilia and flagella. And also I told you it is exclusively present in the animal cells from where the spindle fiber formation take place, and it will give rise to the spindle apparatus, and that is very much important during the cell division. So centriole forms the basal body. Centriole forms the basal body of cilia or flagella. It forms the basal body of cilia or flagella. In animal cells, centriole give rise to spindle fiber, which in turn form spindle apparatus, which play a very vital role during cell division of animal cell. Okay, so this is the function of the centriole, we can say. So now it should be clear to you that it is present only in the animal cell. In plant cell, it is absent. So you may be asking then how the cell division takes place in the case of animals, in the case of plant cell, as the centriole are absent. So in them also the spindle apparatus formation take place, but that do not occur with the help of centriole as it is absent. So that will occur by another method, and that I will discuss in that particular topic. Okay, so this completes the centrosome and the centriole. Clear? Now one more topic, one more cell organelle is remaining, that is the most important one, that is the nucleus. Clear? And about nucleus I will start in the next video. Thank you.